third line of defensive mechanisms, the peculiar characteristic is the immunity is specific. That means this immunity is responsible for differentiation between self and non-self. Self means body substances and non-self means the foreign substances. These foreign substances are called as antigens. So we have seen that specific immunity is divided into two categories: humoral immunity and cell mediated immunity. So, in order to understand this immunity, that means whenever any foreign substance enters in our body gives the response in the form of production of antibodies and production of sensitized T cells. Production of antibodies and sensitized T cells is called as immune response. So, immune response involves production of antibodies and production of sensitized T lymphocytes. This means that the type of white blood cells mainly involved in third line of defense are T cells and B cells. So, in order to understand the immune response, let us see what are the organs which are involved in the immunity or immune system. These organs, as we have seen yesterday, are divided into two categories: primary lymphoid organs and secondary lymphoid organs. The function of primary lymphoid organs is synthesis of white blood cells, red blood cells, or thrombocytes, and maturation of T lymphocytes. That is the function of primary lymphoid organ. In our body, we have two primary lymphoid organs. First one is bone marrow. Bone marrow is the tissue that fills all the cavities of long bones, which is responsible for the synthesis of blood cells. Thymus is another primary lymphoid organ which is responsible for the synthesis of T lymphocytes. That means B lymphocytes are produced in bone marrow and T lymphocytes are in thymus. Then secondary lymphoid organs are those in which immune response is developed. That is, there is production of antibodies and production of T lymphocytes. So there are many secondary lymphoid organs which are situated in our body, which we have seen yesterday. So that is the site for generation of immune response. Humoral immunity we have been discussing yesterday. It is B cell mediated immunity. So there are millions of B cells we are having in our body. Whenever any foreign substance enters in our body, a specific type of B cell, it identifies that particular antigen with the help of receptor, and this specific type of B cell undergoes activation. Further, it undergoes proliferation and then differentiation into plasma cells, which then secretes antibodies. Some of these B lymphocytes get differentiated into memory B cells. Further, these antibodies are responsible for several functions. That means, how exactly these antibodies are responsible for elimination of foreign substances or microorganisms. So there are three functions of antibodies. First one is neutralization. Antibodies bind to pathogens, toxins, viruses, bacteria, protozoa, fungi, and many more different types of organisms, and it blocks its activity, like it blocks its entry into several tissues. It prevents or it eliminates its further growth or entry into the different tissues. That is why it is called as neutralization. Here you can see in the diagram, virus is being bound by antibodies around, so that Now this virus doesn't have sites for its binding to the different host cells. This is how the virus is neutralized. Then a toxin produced by bacterium Clostridium diphtheria is called as diphtheria toxin, which is responsible for life-threatening symptoms. So this toxin is, if the antibodies are there, these antibodies will bind to this toxin and it will prevent the activity of this toxin. This is how the neutralization occurs in case of other antigen. Second one is antigens bound by antibodies are easily engulfed by phagocytic cells. So when antigen binds to antibodies, then the complex is called as antigen antibody complex. Such complexes means after neutralization, these antigen antibody complexes can be easily engulfed by phagocytic cells and further leads to their destruction, as we have seen in case of phagocytosis. This process is called as opsonization. This opsonization is like this. You can see that this is antigen antibody complex. Pathogen is trapped by antibodies and further this antigen antibody complex is then engulfed by macrophages that leads to their further destruction. Third function is activation of complement pathway. We have seen that complements are lytic proteins which are responsible for the killing of bacterial cells. So antibodies bound to the antigens activate the complement pathways that cause lysis of microbial cell. That means these antigen antibody complexes are also responsible for activation of complement. So these lytic proteins, they arrange on the bacterial cell wall and then they lead to formation of holes or pores. As a result, whatever the nuclear content is there, cytoplasm, it is released out. Ultimately, that leads to the death of organism. These are the functions of antibodies. These are the functions of humoral immunity. Second type of immune response or immunity is cell-mediated immunity. In this case, T cells, T lymphocytes are involved. So there are several classes of T cells. First one is Th cell, which is called as T helper cell. So the function of T helper cell is these cells, they help B cells for the production of antibodies. We have seen the process of production of antibodies by B lymphocytes. The initial step is B cells first recognizes the antigen and later on the second step is its activation. So this activation process, it requires help from T lymphocytes. Suppose this is the B cell which has recognized an antigen and further it needs to be activated by T lymphocytes like this. So these T helper cells, they bind to this B lymphocyte. We call this B lymphocyte as sensitized B lymphocyte. As a result of that, these T cells, they secrete chemicals and these chemicals are responsible for activation of B lymphocytes. So this is how after the release of cytokines, B cells, they get activated. After activation, it undergoes proliferation and further differentiation, as we have seen earlier, into plasma cells, and then these plasma cells secrete antibodies. This is how the role of T helper cell can be explained. Second type of T lymphocyte is cytotoxic T lymphocyte, abbreviated as TC. It is responsible for the killing of virally infected cell. Nowadays in COVID-19 situation, we should be able to understand the role or importance of this TC cell in the killing of uh, coronavirus infected cells in the respiratory tissues. This is how it happens. So this is the target cell. Say for example, this is the cell which is infected with viruses. Virus is an intracellular parasite, so it grows inside the cell. So therefore antibodies are not useful in order to kill these viruses, because these viruses are hidden or they are trapped inside the cells. Antibodies cannot enter inside the cell. That is the point to be remembered, that humoral immunity or antibodies are only effective against extracellular pathogens, extracellular antigens, whereas TC cells are effective against intracellular pathogens. So if this is the target cell which expresses uh, certain receptor molecules on these cells, for example class 1 MHC molecule, this is then bound with the help of receptors. It is bound by the TC cell. TC cell, it is a type of CD8 cell. So as a result of binding, this TC cell releases the enzymes or the contents onto this virally infected cell, that leads to destruction of this cell, and that leads to the formation of pores. And this is how, after the killing, the TC cell is released and the infected cell, it dies. That is the role of cytotoxic T lymphocytes, which is important in viral infections. Another one is TD cell, T delayed type hypersensitivity cell, which is responsible for activation of macrophages for the digestion and killing of ingested microbes. Sometimes during the process of phagocytosis, the initial steps are attachment to the foreign particle and engulfment. After engulfment, there is formation of phagosome. Then the next step is phagosome should fuse with lysosome. But sometimes for this fusion process, these macrophages needs activation, and this activation is given by TD cells. So that is the role of TD cells, activation of macrophages for active phagocytosis. Suppose this is the macrophage. We can see the phagosome in which red colored wire shaped organisms are there, and in white vacuole, then you have these red colored vacuoles, these are lysosomes. So already we can see that this macrophage has engulfed the microorganisms, but the next step hasn't been happened. So in order to perform the next step, that is fusion of phagosome to lysosome, these macrophages needs and assistance from, you can see this is the phagosome, it needs assistance from the another type of cell, that is TD cell. So TD cell, it is also abbreviated as TH1 cells. So as a result of this TD cell, it releases chemicals called as cytokines, and these cytokines causes the activation of these macrophages. Activation leads to the, now you can see the fusion of phagosome with lysosome. This is how TD cells help the macrophages to digest the pathogen. Then the next one is T suppressor cell. T suppressor cells are involved in the regulation of immune response. So whatever we have seen so far, role of B cells and role of T cells. So there has to be some regulation. That means once the production of antibodies starts, how long it should continue? It should stop after some time, or how long the production of TC cells continue, how long the production of TH cells continue. So there has to be some control mechanism. To T suppressor cells, as the name indicates, it suppresses the immune response. That means they are involved in regulation or control of immune response. That is about humoral immunity and cell-mediated immunity. Next part that we study in immunology is immunological disorders. Immune response, we have seen so far, is beneficial for the removal of pathogens. Sometimes this immune response may be harmful. If this immune response is raised or generated against our own body cells, then this condition is called as hypersensitivity. This is also called as allergy. Sometimes this immune response may not be generated against the pathogen, and this is called as immunodeficiency. That means there is no resistance power, so that leads to immunodeficiency. So hypersensitivity or allergy, it is the immune response generated against harmless antigens. That means it is not generated against the pathogen. The immune response is generated harmless antigens. Say for example, the chalk powder or the dust particle that we inhale. So sometimes we may get allergy to that particular dust powder, or sometimes there may be allergy of penicillin antibiotic. So penicillin is not a pathogen, penicillin is harmless, but sometimes in some individuals, injection of this penicillin, it leads to generation of immune response, and that immune response is harmful for our body. So for examples, these types of harmless antigens are dust particles, drugs, foreign proteins, plant materials, and animal products. The examples of allergic diseases are asthma, farmer's lungs, and hemolytic disease of newborn, tuberculosis, and leprosy. Immunodeficiency means inability of person to generate immune response that protects us from pathogenic microbes. In this case, the person may not be able to produce antibodies, T cells, or both. Such individuals are highly susceptible for infections. Then another section in immunology is diagnostic immunology. As the word indicates, diagnostic. This means that there is use of antigens and antibodies in the diagnosis of diseases or infections. So immunologic reactions means antigen antibody reactions. One point is very important that we have learned. Third line of defensive mechanism is specific. This specificity is due to the specificity between antigens and antibodies, antigens and T lymphocytes. Specific type of antibodies can bind to only specific type of antigen. For example, antibodies generated against poliovirus will only bind to poliovirus only. It will not bind to some other type of antigen. That is called as specificity. So this specificity is very important in the diagnosis of diseases. There are two types of reactions which are useful for the diagnosis of diseases, and these reactions are two types of reactions that occur between antigen and antibody. If the reaction is visible, so these two reactions are precipitation and agglutination. So there are various types of antigens. Antigens means disease causing microorganisms, bacteria, fungi, protozoa, viruses, and different types of antibodies. These are the antibodies which are produced after the entry of pathogen in the body. So antigens and antibodies both are the indicators of infection. So if the person is infected with coronavirus, then there are two possibilities: that the person is having those viruses in his or her body. That means there is presence of antigen. And the another possibility is following the entry of this coronavirus, then it leads to the formation of antibody. So the person who is exposed to this coronavirus infection must be having antibodies in his body. So the indications are both, whether the person is suffering from COVID-19 or not, either presence of antigens or presence of antibodies. So both can be detected by using this diagnostic immunological kits. First reaction is precipitation reaction. We are not going into the too much details of precipitation reactions. And second one is agglutination reaction. Both these reactions are useful for detection of antigens and antibodies. These are the several examples of agglutination reactions: hemagglutination, ELISA, RIA, fluorescent antibody technique, complement fixation test. The tests which are based on antigen antibody reactions are called as serological tests. Serology means study of serum. Serum is a component of blood. If you collect the blood in means after the collection of blood outside the body, then if you collect it in a vial, after some time what will happen, that blood will coagulate, and after coagulation small amount of fluid which is released out of this clot, that part is called as serum. So it is a watery part of blood, that is serum, and this serum contains antibodies and the antigen to be detected. So that is why this is called as serology. So it is, what is done in serology is detection of antigens with antibodies, or detection of antibodies with standard antigen. This is very important. Detection of unknown antigen with standard antibodies, and detection of antibodies with standard antigen. Both ways are possible. Patient's blood or tissue or swab is collected and it is mixed with test reagents. So I'll give you the reference of current antigen based test for the detection of coronavirus infection, COVID-19 disease. So this is a very fast test. So within few minutes you will get the result. So that antigen based test is like this. So what is done, you have seen that the swabs is being collected from the patient's nasal cavity. Then that swab is immersed into saline water, and then that saline water is ready for the antigen test. Or sometimes there may be collection of blood, so either it may be blood or the swab saline. So what is that swab is supposed to contain, or what is that blood supposed to contain? It is supposed to contain the virus. And these viruses, coronaviruses, are detected with the help of standard known antibodies. We call it as test reagent. So if there is presence of this virus in the sample, it will be bound by antibodies and there will be antigen antibody reaction. If there is antigen antibody reaction, then this indicates that there is presence of coronavirus, and therefore we can diagnose that the patient is infected with coronavirus. Another example is detection of hepatitis A. So generally hepatitis A is diagnosed by identifying antiviral antibodies present in the body. So this is the example of diagnosis of hepatitis A disease by identifying antibodies with standard antigen. This is the opposite case. So both ways are possible. On the other hand, hepatitis B is diagnosed by identifying viral antigens from viral code. So that is what happens in case of COVID-19 diagnosis. We can see the example of this kind of reactions. See this, that patient's serum is being collected. That means the patient's blood. So that patient's blood may contain viruses. So we call them as viral antigens. Then this sample is mixed with standard antibodies, so which are already purified, and we have that in our laboratory in pure form. Standard antibodies. These standard antibodies are known to bind to specific antigen. So when this is mixed with antibodies, then if the antigen is present, antibodies are going to attach or bind to this antigen, and there will be formation of antigen antibody complex. Formation of antigen antibody complex, it indicates that the patient is infected with virus. Second type is diagnosis with the help of antibodies. So in the similar fashion, patient's serum is collected, which is supposed to contain antiviral antibodies, and then, like earlier one, it is mixed with standard antigen. In the earlier case, it was detection of antigen with the help of standard antibodies, and this is the case in which detection of antibodies with standard antigen. So it is mixed, and if due to specificity, if there is formation of antigen antibody complex, then we can say that the patient is suffering from the virus infection.